Namaste students, Pushpa Vidya. I, JP and Dr. Vaichandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the chromosome. Chromosomes are the thread-like structures located inside the nucleus of animals and plant cells. Each chromosome is made up of proteins and a single molecules of deoxyribonucleic acid. Okay, it is called DNA, which is passed from the parents to the offsprings. Okay, so DNA contain a specific instruction that makes each of the living creature a unique. Okay, that means how a person differ from each other. That is determined by the DNA. Okay, of a chromosome. So the relative positions of a chromosomes in a cell, that is, first let's discuss about the history, a brief history of chromosomes. Chromosomes are the rod-shaped, dark-streamed body seen during the metaphase of a mitosis. So during mitosis phase, we can see the chromosome in a prominent form. The Strassenberg discovered chromosome in 1875. The term chromosome was coined by Vallier in 1888, okay, which was initiated as chroma means color, okay, and soma means the body. Okay, so the unique features of a eukaryotic chromosomes are the chromosomes are best visible during the metaphase. Okay, so the chromosome bears the genes in a linear fashion. That means the chromosomes vary in shape, size, and number in different species of the plant and animals. Chromosomes have properties of self-duplication and mutation. Chromosomes are composed of DNA, RNA, and proteins. Okay. So when we see the size of a chromosomes, it is measured at mitotic metaphase. Generally, the measures in the length or the diameters. Okay. So the plant usually have the longer chromosomes than animals. Plant chromosomes are generally of 0.8 to 7 micrometers in length, whereas in the animal it is 0.5 to 4 micrometer in length. So the chromosome size vary from species to species depend upon the which type of species it is. Okay, and the chromosome shapes are generally determined by the position of the centromere. Okay, and the chromosome generally exists in three shapes, that is rod shape, J shape, and V shape. When we talk about the chromosome number, each species has a defined and a constant somatic and genetic chromosomes. Okay, so that means the somatic chromosome number is the number of chromosomes found in the somatic cells, while the genetic chromosome number is the number of chromosomes found in the genetic cells, gametic cells here. Okay, that means the somatic chromosomes is always diploid and not donated as a 2n, whereas the genetic chromosomes are generally haploid and denoted as n. That is the haploid form. Okay, so here the in human beings, the somatic chromosomes are X, X, and XY, whereas the genetic chromosome number is X and Y. Okay. So here, when we see the different somatic number or the of uh, in plants, like and animals, like Homo sapien, which we call that as human, they have the somatic chromosome number is 46, but whereas the gametic number is 23. In Oryza sativa rice, where it is 24 and the gametic number is the 12. Okay. Allium cepa, which we called it as onion, and the somatic number is the 16 and its gametic number is the age. Okay, so the chromosome structures, when we see, it consists of the several parts like centromere, chromatid, secondary construction, and the satellites, telomeres, chromomere, chromatinema, and the matrix. Okay, so these are the seven parts of when we have to see the structure of any chromosome. These structures determines uh, completely about the chromosome. Okay, so let's me share the PPT. Yeah. So let me share the PPT image. So here, so in the structure of the chromosome, you can see the presence of the telomere. In the central part, it is the centromere and the chromosome. These are the chromosome arms. Okay, and this complete structure is called chromatid. And these are the small ketocores are present on the adjacent side of the uh, two arms. Okay, so number one, first we have to see that the centromere is the localized region of the centromere chromosome with which the spindle fibers are attached and is known as the centromere or the primary constriction or the ketocores. Okay, so the chromatid is one of the two distinguished longitudinal subunits of the chromosome and is called the chromatid. So chromatids are of two types, that is the sister chromatid and the non-sister chromatid. The secondary constriction, that is some chromosome exhibit secondary constriction and is added to the primary constrictions. So the chromosomal region between the telomeres is called the satellites or the transplant, and the chromosomes having the satellite, which we call that as the satellite chromosomes. Okay, so the telomeres are the two ends of the chromosomes and they are called the telomeres, and these telomeres are highly stable. As we have seen in the diagram, they are not fused or united with the telomers or other chromosomes. Okay, so here the chromomere is the some specialized and show small bead-like structures and are called chromomere. So the structure of the chromomere is the chromosomes and is constant. Okay. So the chromonema is a thread-like coiled structure found in the chromosomes and the chromatid and is known as the chromonema. So the next is the matrix, and matrix is the fluid part and is called the chromonemata and are embedded in the matrix. Okay. So here when we see the chemical composition of a chromosome, so the chemical composition of a chromosomes are the nucleoproteins nature and the nature, or which means that the RNA, DNA and the protein. Okay. So generally the chromosome contains 30 to 40 DNA or 50 to 65 protein and 0.5 to 10% of the RNA. So the DNA, the amount of DNA present here in the somatic cells is constant. So the DNA constant of the genetic cells is half of that of the somatic cells. So the DNA of the chromosomes of the two types, that is the unique DNA or the repetitive DNA. So the unique DNA or the unique DNA type consists of those of the DNA sequence which are present in a single copy of a genomes and are unique in nature. Okay. So the unique DNA of or also known as the repetitive DNA or the coding of a protein, which requires a large quantity of the cells for the storage of the protein. So here we have to see that what different unique DNAs are also known as non-repetitive DNA. So the code for the protein are unique in large quantity for the cells and the storage proteins. So here the repetitive DNA or the repetitive DNA consists of the DNA nucleotide or the base sequence, which are few in nature in the base and long, and they are present in several to the million copies. Okay. So per genomes or the human genome contains 30 to 30% of the repetitive DNA, or the repetitive DNA is further divided into highly repetitive DNA or number two moderately repetitive DNA. Okay, so that is the repetitive DNA, and second is the RNA, which is a purified chromatin content of 10 to 15% of the RNA. That means RNA associated with the chromosome is the messenger RNA, and the RNA transfer and the ribosomal RNA. Okay, so here the protein part is associated with the chromosome is also classified into two broad groups. That means number one is the histone, okay, and the number two is the non-histone proteins. So number one, the histone proteins, and the number two is the non-histone protein. Okay, non-histone proteins are acidic in nature, and histone proteins are basic in nature because of the basic amino acids. Okay, so here the histone protein constitute about 80% of the total chromosomal proteins. So here we have to see the presence of almost 1 to 1, that means 1 is to 1 ratio of the DNA, or the 5 fractions of the histones are present, like 1H1, 2H2, okay, 2H3. So non-histone proteins are the non-histone proteins which are made up of 20% of the total protein mass. Okay, that means that content of the non-histone protein is different from the species to species, and the non-histone protein includes many important enzymes like DNA and RNA polymerase. Okay, so when we see that two types of chromosomes, according to the relative positions of the chromosomes, are divided into four parts. That is centromere location, metacentric, subcentric, submetacentric, acrocentric, and telocentric. Okay. Next is the karyotypes. That means the complete set of chromosomes in a species or in a individual organism. We call it as karyotypes. So the basic number of the chromosome in a somatic cell of an individual or a species is called as the somatic number, and which we can designate here as 2n form. So here thus we can see that human chromosome is 2n, that is the diploid form, and this diploid form has 46 number of chromosomes, that is 23 pairs. Okay. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any queries, we'll discuss in the problem solving session. Thank you.